Perception is a special kind of problem-solving process. The problem of visual perception is how to represent the world of objects and events around us based on the photons of light falling on the right retina at the back of the eye. We will focus on vision because it is a dominant sense in primates and its first basic properties are well understood. Cognition and action begin with perception, and if we are to understand how the brain works, we first have to understand how the brain perceives the world around it. In this presentation, we focus on the visual pathway from the eyes to the first stage of visual processing in the occipital cortex. The first step in the visual pathway takes place in the interaction between the environment and the eye. Due to the optics of the eye, images are inverted on the retina at the back of the eye, and they are focused using the lens of the eye. There are two types of photoreceptor on the retina, and each type responds differently to the photons of light that have been reflected off the surfaces of objects in the environment. The rods and cones contain photopigments, which signal neurons further along the visual pathway to fire action potentials. Cones are color sensitive, and they require a high level of light to send signals, making them more useful in the day. Rods do not process color, but they can fire in low light conditions, making them useful at night when we can't see colors because there isn't enough light for the cones to fire. Only about 5% of the photoreceptors in the retina are cones. They see colors in detail, and they tend to be concentrated in the center of the eye in a region called the fovea. When we want to see something in detail, we move our eyes so that the object of interest is in the center of our vision. Thus, we only see a small portion of the world in detail at a particular point in time. By moving our eyes around, we paint the world with a succession of detailed snapshots of different parts of the scene, and we have the illusion that we are seeing everything around us in full detail. Many experiments have demonstrated that our feeling that we are seeing everything in the world around us clearly all the time is in fact an illusion. A lot of processing of visual information occurs in the eye, and there is a lot of information compression. By the time it leaves the eye, the many millions of photoreceptor activations have been compressed and transformed into 1 million fibers that exit the eye as the optic nerve. The layers of the retina can be clearly seen in highly magnified microscopic views. At the top of the visual processing hierarchy within the eye, the ganglion cells send activation through the axons of the optic nerve. There are about 2 million ganglion cells, so each ganglion cell receives information from several receptors. Ganglion cells connected to cones have high acuity, meaning that they can see fine detail. The optic nerves from each eye cross over in such a way that information from the left visual field of each eye travels to the right side of the brain, and information from the right visual fields of the two eyes travel to the left side of the brain. The point at which the optic nerves from each eye meet and then have this crossover of visual fields is referred to as the optic chiasma. Note that the information about which part of the visual field things have been seen in is retained. This retinotopic mapping travels all the way to the visual cortex, where different parts of the cortex handle different parts of the retina. On the way from the optic chiasma to the visual cortex, the visual information stops off at the lateral geniculate nucleus of the thalamus, where the retinotopic layout is retained in different layers of the lateral geniculate nucleus. The thalamus is an important way station deep within the brain and just above the brainstem. The architecture of our visual system is ancient, and it is shared with other mammals. The visual pathway is not just some abstract thing that researchers have constructed from data, it is something you can dissect in a dead brain. Even the radiations of nerves from the lateral geniculate nucleus to the visual cortex and the associated retinotopic mapping can be clearly seen. The wiring in the brain is a lot like the electrical wiring within and between devices. The brain is full of wiring. The wiring for visual perception is relatively easy to understand, but it becomes harder to interpret the wiring when the information is processed at a more abstract level and there is no longer an obvious connection to basic information such as that contained in the retina. Every cell in the visual processing hierarchy refers back to the information that was received at the retina. Receptive fields contain either inhibitory or excitatory centers with surrounding areas that are in the opposite. When a spot of light is shined on the center of an on-center ganglion cell, the neuron's firing rate increases, while firing decreases when a spot of light is shined or shone on the surrounding area. The off-center cell acts in the opposite way. When a spot of light covers both areas equally, the inhibitory area and the excitatory area cancel each other out, and the ganglion returns to its baseline firing rate. This makes ganglion cells effective at determining contrast between objects in the environment and forms a basis for later visual feature recognition in the visual cortex. Cells have baseline firing rates, so if you stimulate a cell, for instance by having the center of an on-center cell activated, then the firing rate increases. Conversely, if the surrounding part of a center surround cell is activated with light, then the firing rate decreases below the baseline level, but it increases again when the light is switched off. When light covers both the center and the surround, these two activations tend to cancel each other out, and you get different patterns of firing depending on how much of the center versus how much of the surround is covered. Center surround cells gather information from multiple receptors. For instance, information from 3 receptors may be aggregated into a center, and information from 4 receptor cells might be aggregated into the response for the surround. Simply by combining inputs from different receptors and varying the sign, visual features can be constructed. Perception is not passive. When we see, we have to plan our eye movements based on what we are seeing. This is why 10% of the visual information is sent to part of the brainstem to assist with eye movement planning. Another demonstration of the active nature of perception is that the number of fibers coming into the lateral geniculate nucleus from the visual cortex is greater than the number of fibers coming from the eye. Some of this feedback input from the visual cortex is used to control attention, and feedback from the cortex may also be involved in inhibiting visual information when it is not needed, e.g. when sleeping. This has been shown when people's eyelids are opened while they are sleeping. The feedback from the cortex becomes more active and they do not actually see. Detailed perception of visual information from the eyes occurs in the visual cortex located within the occipital lobe of the brain. One key point is that visual processing is fast. This makes a lot of sense in an evolutionary context, since in a hazardous environment, actions are often needed to be performed very quickly after something is perceived.